So broadly, today I'm going to talk about some of our work looking at the consequences of chromosomal instability in high-grade serous ovarian cancer. So I like to think of, and others do too, think of chromosomal instability as a process or set of processes that cause the ongoing accumulation of large genomic changes. What do I mean by large genomic changes? Here I'm talking about structural variants and copy number alterations. For the purposes of my work and the talk today, I mean pretty much everything. So everything from deletions and duplications through to more exotic changes, so inversions or insertions or rearrangements, as in addition to what we think of as complex structural variants, so combinations of simple structural variants is easy enough way to think about them for the purposes of my work. And these sort of large changes are the changes I'm thinking about in terms of the changes that happen to our genomes as a consequence of CIN, or chromosomal instability. So I'm going to highlight two stories today. Typically we think about the processes of chromosomal instability causing the genomic changes. In the first story I want to talk about, I'm going to explore the possibility that perhaps these larger genomic changes could be driving chromosomal instability. That might be the case in the case of BRCA1 and 2 alteration and homologous recombination repair deficiency. And then I also want to talk more broadly about what the diversity of structural changes in high-grade serous ovarian cancer looks like. So the more conventional, we know these tumours are subject to multiple processes of CIN. What do the consequences look like in these tumours in greater detail than we knew before? Okay, so thinking about high-grade serous ovarian cancer, I'm sure many in the audience are very familiar with this cancer type because it's a common one for looking at structural changes in. But I want to highlight a few things. Firstly, it's a cancer type that's not driven by recurrent mutation. However, we don't understand fully what the mutational landscape looks like. Our understanding is fairly incomplete, but we do know that it's driven by copy number changes with the exception of P53 mutation. That said, one of the biggest success stories in our ability to target chromosomal instability is the application of PARP inhibitors in tumours that have short variants, so single nucleotide variants or indels, either inherited or occurring in their tumours at BRCA1 and 2. And these patients benefit hugely from PARP inhibition treatment. The plot on the right there is from the SOLO-1 trial, where patients with these changes were treated either with olaparib, so a PARP inhibitor, or placebo, and the PARP inhibition results in a 70% reduced risk of disease progression. So the effect of these treatments are huge for these patients. And these are patients with mutations, to be clear, not large structural variants. But we don't understand potentially which patients will benefit from these treatments. Our expectation is about 50% of high-grade serous ovarian cancer patients may benefit from PARP inhibitors, but beyond those that have BRCA mutations, potentially BRCA1 methylation, it becomes a fuzzier picture. So that's an open question for the field. What we were particularly interested in is how we begin to understand the rest of that pie chart beyond the patients with mutations. So a few years ago, myself together with Colin Semple's group and Charlie Gurley, as well as some industrial partners, so AstraZeneca and Precision Medicine Scotland, and the Scottish Genomes Partnership, brought together a large combined cohort of whole genome sequences from high-grade serous ovarian cancer patients. These sequences, some of them were available before and published before, but we brought them all back to FASTQs and reprocessed them in a uniform way to create this uniform call set. We also sequenced an additional over 100 Scottish ovarian cancer patients to supplement the existing sequences. We also, on top of the genome sequencing, so on top of whole genome sequencing, we have paired RNA-seq for many of these cohorts. So for the Australian cohort in TSJ existed, and the MD Anderson cohort existed already, but we also did matched RNA-seq on the same piece of tumour for many of the Scottish samples. So it's a valuable resource. We characterise these tumours in lots of different ways. I'm only going to talk about a couple of them today, but that data is there, and expect to see many papers coming out on it in the near future. Okay, so thinking about this first question, not just are these large genomic changes consequences of CIN, could they potentially cause CIN? So perhaps low-hanging fruit or easy question to think about in the context of high-grade serous is we know short mutations, so single nucleotides and indels, cause homologous recombination repair deficiency. But what about structural variants? What happens if these genes are altered through deletion or other larger changes? With our whole genome sequencing, we were able to look at that. And what we saw is that we do see many large structural variants at both of these genes, and they're a frequent source of disruption. So only looking at 3 cohorts here, because these were the ones we had the sequencing data and QC'd and done all the full pipeline on at the time. We saw that these genes are frequently altered, and the alterations tend to be very big. So what surprised us is the deletion of BRCA1 is very common and has a median length of about 5 megabases. So these are huge changes. And we see a very similar pattern at BRCA2 as well, a slightly increased rate of duplication, which is interesting, I think. But the pattern of deletions is broadly similar. We're then able to ask, are these changes just happening there by chance? These tumours are very messy. Are we just seeing deletions like this everywhere? And we're able to show that these 2 genes are being deleted more often than the rest of the genome. So that gives us additional evidence that these are not just being deleted by chance. That may still be the case because this is observational data, but it's reassuring. What's even more reassuring, I think, that these deletions are doing something and worth considering is that with our matched RNA-seq data, we were able to directly compare the BRCA1 expression and BRCA2 expression in the presence and absence of these large deletions. The plot on the left here, each bar here is a patient or a tumour, and they're coloured according to the presence of deletion. The easiest thing to take from this is that the blue bars have deletions. The grey have nothing. On the right, we can see the level, and the height of the bar is the level of expression of the BRCA1 gene. On the right is the same data presented as a box plot, and you can see that the expression of BRCA1 is much lower in the presence of deletion. We thought that was incredible that you could see that from noisy patient bulk RNA-seq data. We didn't expect to see anything. RNA-seq is subject to many different confounding factors, and we were really surprised that we saw it differentially expressed these genes with differential levels of expression in these data. We see the same for BRCA2. So really, this is just adding to our hypothesis that these deletions are reducing the function of these genes. Now, getting back to CIN, so we know about 50%, or expect about 50% of high-grade serous ovarian cancers to be homologous recombination repair deficient. Many others have developed methods and tools to use whole genome sequences and less than whole genome to predict whether a given tumour is HR deficient based on its genome, based on the scarring. These are some of them. There are also more. I'm sorry, Florian, yours is not here. But there are many. I do not choose between them, but you can use them. And we've used them on each of our tumours to get a readout from the genome of how likely a given tumour is to be HR deficient. And then using that readout, we're then able to compare the readout according to what's going on somatically at BRCA1 and 2 in terms of large genomic rearrangements. So what we saw is that, as you would expect, those tumours with short variants, so single nucleotide variants in indels, the ones we know are HR deficient, or as much as possible we know are HR deficient, they are predicted to be HR deficient. Hooray! But what we also saw, and it's small numbers, but bearing in mind this, at the time at least, is the largest collection of full genome sequences from this cancer type, the tumours that had deletions at both BRCA1 and BRCA2 in the absence of single nucleotide variants or indels, so they've got nothing else going on at those genes as far as we can tell, the patients that had deletion of both genes also were predicted to be HR deficient. That's not been shown before, and I think is really interesting for two reasons. One, it identifies new patients that may benefit from PARP inhibition that are not currently getting it. And two, I don't know that we appreciated that tumours were likely to delete both of those genes. We don't generally see that for short mutations. If one gene is mutated, it doesn't tend to be the case that the other gene is mutated as well, because that doesn't seem to be the case for deletion. For those tumours that just had a single deletion of one of the genes, it was a messier picture. So we still have to understand there what else is going on. Probably a single deletion is not sufficient, but what else do you need for those tumours to be HR deficient? And we don't know the answer to that. For the rest of the events, we do see lots of other events. So I'm talking about deletions here, but we did look at the full range of potential changes that could occur. So we do see a reasonable number of inversions and duplications and more complex things too, but here in fairly low numbers and a messier picture, so we don't say too much about them. What we then sought to ask is, so in ovarian cancer, the rate of point mutations is pretty high. So these patients are already being treated, and the rate of rearrangement is maybe a bit lower at these genes. But in other cancer types, those patients are not getting PARP inhibitors at all because their rates of mutation are so low. But do they have larger changes, and therefore that might be a cancer type where this sort of targeted treatment would be beneficial? We're not looking there because they don't have the point mutations. So we looked at the PCOG database and looked at the deletion status of BRCA1 and 2 across all the cancer types that had enough tumours. And one thing that stood out is that it's very common. And in particular, soft tissue leiomyosarcoma, for example, 50% of tumours have BRCA2 deleted. That doesn't necessarily mean they're HR deficient, but it means these changes are happening a lot. So we need to understand what the impact of these are. We were also able to look at expression in other cancer types. And like we see in our ovarian cohort, BRCA1 deletions in particular do cause a differential, a significant decrease in expression. For BRCA2, the expression is also lower, perhaps to a lesser extent, but I think it's a bit messier there. But certainly the fact we're seeing this signal in across cancer types in bulk tissue is incredible. So to be clear, I think this is exciting. I think the deletions of BRCA1 and 2 could be causing HRD. I'm not saying they are causing HRD from this. We've gone on to experimentally validate them in the lab and are looking at it in other cancer types. So I would just say watch this space, but that could be exciting for identifying patients that may benefit from treatment. Okay. I want to talk about the second part of my talk now. So the more classical way of thinking about chromosomal instability processes is to think our tumours are under these processes that are causing large changes, and to look at the changes as a consequence of those processes. And those two tend to get pushed together so closely that we think of them as one and the same thing. I'm going to talk very much about the consequences here rather than the processes, because I think by we need to look at both ends. I'm one of the people that looks at the end point, at the patient tumour, which is messy and full of the changes that you see at the end result, rather than piecing together how they got there. In high-grade serous ovarian cancer, we haven't worked out what drives these tumours exactly. This is where we were at in 2023. About half of the pie chart have mutations in HRD-related genes or expected to be HRD. But the other half of this pie chart is the half I want to talk about now. Very much don't really have any recurrent mutations. There are one or two genes that have mutations in, but they're not being driven by mutation. They're being driven by copy number changes in structural variants. But we don't understand exactly how many, what the rates of those things are, where they're happening, what sizes are they. It's only through our large whole genome sequencing cohort and others that we can detect all of these changes and begin to firstly describe and then understand what the consequences are of those changes and where those changes have come from. So looking both ways at the functional and clinical consequences, looking forward and looking backwards at how they got there, at the mechanisms of CIN that have generated them in the first place. So we sought to characterise what this full picture looks like using the whole genome sequencing. And it looks messy. So I'm going to start by talking about complex SVs again. Because by messy, what I mean is there are rearrangements and changes, copy number alterations everywhere. They're not happening in isolation. There's not, oh, we see this change here and this change here. Anyone who's looked at a ovarian cancer genome or sarcoma genome or many of the other cancer types that are also very rearranged will know they look incredibly messy everywhere. So how do you begin to disentangle what that means? And one of the ways we as a field have begun to do that is to characterise what particular combinations of structural variants mean according to what we expect the mechanism is that's generated them. So some of these we're terming complex structural variants. So some you'll have heard of before, or we've given names to before. And these include chromothripsis, so genome shattering. Here the hallmarks are fairly well published and well known, and we can predict them from whole genome sequencing and say with reasonably high confidence that we think chromothripsis has happened in this place, in this tumour. Others, so like famously the tool Shatter-seq, developed by Cedro, but also other tools. We can also detect breakage fusion bridge cycles, chromoplexy, a few others, so Pergo, Rigma, Typhonus, ecDNA. Lots of people have developed methods for detecting ecDNA from whole genome sequencing, but also layering on optical mapping, for example. So this is a fairly hot topic, and new methods of detecting these events in whole genome sequencing are emerging all the time. But we don't understand what the consequences are of these things. We don't know how often they're happening really in many cancer types. It's been reported previously, but in fairly small numbers. So there's a lot to learn here. So the first thing we thought we'd do is to detect all of the varieties of these things that had been published at the point that we had sequencing, and ask how often they're happening, where are they happening in our tumours. So that's the data I want to share with you just now. So this is work by Stuart Brown, who's a PhD student in the lab, and he used Shatter-seq as well as the JABA method to detect all these various different types. He also used Amplicon Architect to detect ecDNA, compared the rates between different tools, and we eventually got to this set of prevalences. And what you can see is that the prevalence of all these events is really high, not unexpected given the messy rate of these genomes. The top two are HR deficiency, around about 50%, not a complex SV, but there is a kind of a benchmark, and whole genome duplication, again, about 50%. But all of these things, including the ones that are previously thought to be quite rare, are happening at high rates in our tumours. The exception maybe is Typhonus, which is pretty rare in high-grade serous ovarian cancer. I'm not sure if anyone's seen that elsewhere. I'd be interested if they have. I think it's more common in other cancer types. And one of the things that we saw is that these events across the board tend to all occur in whole genome duplicated tumours. That makes sense. It makes sense if you have additional copies of your genome, then perhaps you've got a higher tolerance for breaking it in some way. So shattering it or holding this load of mess. If there's extra copies, then presumably cells can still function if you break them in this many ways. You can tolerate this amount of instability. What we didn't appreciate as much was that the HR deficient tumours don't tend to be those tumours. So there is this anti-correlation between whole genome duplication and HR deficient. It's not a perfect mutual exclusivity, but our tumours, as you'd expect from the pie chart potentially, is that the messy, rearranged tumours tend to be not the ones that we can target with PARP inhibitors, tend to be the ones that we don't have good treatment options for. If we look at this in more detail, here we have, instead of just whether a tumour has been whole genome duplicated, or has been totally duplicated, we have the fraction of the genome that is duplicated. All these measures are predictions, so looking at the fraction of genome duplicated is like a slightly more nuanced measure than asking whether whole genome doubling has happened or not. And we can see that in the tumours with more of their genome that's been duplicated, those tumours that, fitting our hypothesis, tend to be more likely to have breakage fusion bridges and chromosynthesis than those that don't. On the flip side of that, the ones without breakage fusion bridges or without chromosynthesis are fairly bimodal. So some of them are whole genome duplicated still. So what I'm saying here, and this has bearing on directionality, is if you have whole genome duplication, you're more likely to have breakage fusion bridges. But if you have breakage fusion bridges, you're more likely to have whole genome duplication in order to tolerate that potentially. But if you have whole genome duplication, that doesn't necessarily mean you're any more likely to have breakage fusion bridges. So those that are messed up are more likely to be doubled. But just if you're doubled doesn't mean you're more likely to be messed up. I'm going to take a pause. We see that significantly for both of these varieties of messiness, where we have a large amount of data, but it also applies for ecDNA and various others, just the effects are less strong. We're then able, because we've got whole genome sequencing over many tumours, we're able to begin for the first time to look at the genomic distribution of where these events are happening. And in short, it's not random, very much not random. And we see hotspots of complex SVs of every variety on chromosome 19, for example. So you can see this peak at chromosome 19 of all varieties in general, but particularly for breakage fusion bridges, ecDNA, chromosynthesis. And we also see other peaks. So we've got a peak on chromosome 3 and 6, particularly here for RIGMA at chromosome 3. Chromosome 3 has been reported in high-grade serous to be particularly unstable in the past. Whether this hotspot that we're seeing in our messy tumour samples reflects that hotspot that has been reported elsewhere is open for debate and could be investigated. But it's interesting we see this across the genome in signal coming through all the meth. Focusing in on this hotspot on chromosome 19, it's interesting that if we just look at this chromosome, it peaks at a particular point, and at this point is the gene cyclin E1. This is an interesting gene in high-grade serous because we know that overexpression of this gene and amplification of this gene is associated with poorer prognosis. It's mutually exclusive from BRCA1 and 2 mutation, and therefore on that side of the pie chart without feasible treatments. So it's interesting that the possibility that this amplification and overexpression of this gene isn't through simple duplication, but is through more complex mediated events. So the mechanism underlying it may be a more complex mechanism, or a more a CIN-driven mechanism rather than something else. So maybe we do need to be looking at chromosomal instability when we're thinking about cyclin E1 disruption rather than thinking about simple amplification. To test that theory, we specifically looked at the copy number at this gene in the presence of simple duplication versus the presence of all these complex events. And the complex events, as you would expect, were able to amplify the gene to a much higher level than simple duplication alone. This suggests that it might be these complex events might be a mechanism for tumours to achieve these really high copy numbers that they're not able to just through simple duplication. So it might be a reason that this degree of instability is tolerated. We can compare it across flavours of complex mess, and okay, small numbers in some cases, but you see the pattern consistent throughout, and these are all significantly differentially expressed. I'm not presenting p-values, but we've tested them all. They're all significantly higher. Okay, so these complex events are able to achieve higher copy numbers and higher expression of this gene that is linked to poorer prognosis. So you'd expect these events to be good for the tumour, bad for the patient. But that's not actually what we see. If we look at the presence of severe chromothripsis in these tumours, so by severe chromothripsis here, I'm talking the tumours that have chromothripsis in 2 or more chromosomes, so not just one isolated case of chromothripsis. And these patients have better prognosis than others, which is the opposite from what we see, but poses the question of, okay, mess is good for the tumour to some extent, but does there come a point where your tumour has rearranged itself so much and it's subject to so much chromosomal instability that your tumour cells are dying at a higher rate and patients are benefiting more from chemotherapy, for example, and are doing better? And if that is the case, we see it for those patients with severe chromothripsis, but where is that line? How messy do these tumours need to get before it actually becomes better for the patients? And what kind of mess? Shattering here, but what about the other kinds? And is it possible for us to make those tumours messier? Okay, so I'm just going to summarise. I'm not going to talk for the full hour today, and I'll leave time for questions instead. I believe that our genomic consequences of CIN can potentially also be causes, and that knowing what's driving our processes of chromosomal instability may help to target the treatment of our patients, whether those drivers are genomic changes themselves or whether they're other things. But we shouldn't rule out that they could be genomic changes themselves. If we just look at the consequences of chromosomal instability, what we are looking at in real tumours are a huge amount of mess. So we need to find a way to distill that mess into things that can be useful and understood, and get that mess to a point where we understand the functional impact of it and the clinical consequences for patients. At the moment, rearrangements are diverse in size, in type, in impact, but our hypothesis is that they are advantageous to the tumour, but when there are more of them, they could be advantageous to the patient, and we need to work out where that balance is. Okay, and with that, I just want to thank my team, past and present collaborators and funders. Thank you.
Thank you very much for this very inspiring talk. So just a reminder for the audience, so you can ask questions using the Q&A on the chat. Maybe I can start, and this will give time to people to write their questions. So maybe just so one question related to the second part of your talk on the link with between chromothripsis and the better outcome. I was just wondering, so did you check for, potential confounding factors, or did you try Cox regression?
Yes. So that effect, we fitted it in a multivariable model. So we used ElasticNet, and we included signal nucleoside variants, we included copy number changes, we included all recurrent genomic changes that we saw in our tumours. We also included age, stage, HRD, whole genome duplication, everything we had data for that we could think of, and the effects still persist.
Okay, thanks. I do not see any questions yet, then I'll just ask another one. So to the first part, so for the BRCA disruption by structural variants, did you try to time the events? Do you see, whether it's clonal or subclonal? When does it happen in tumor evolution?
So they are deletions for the most part. So we haven't timed the deletions. It would be, as far as I'm aware, not necessarily all that possible from single sample whole genome sequencing. It would be interesting to know whether others have seen that in multi-region sequencing. Do they see the deletions in only one of several regions from the same tumor? That'd be interesting. In general, alterations of these genes are very early, so I would expect that to be true for the deletions as well.
And did you see any examples of such structural variants in the germline as well? Because when we think about the typical BRCA carriers, then we think of the point mutations, but did you see them in the germline?
We looked, we didn't. In short, we saw very little happening in the germline in terms of rearrangement, as you'd expect. But we did look specifically at these genes, and we didn't see any that we were particularly confident in. That doesn't mean that they don't exist, but as far as our callers exist today, we didn't see any.
Okay, thanks a lot. I can't see. Yes, no. Okay, so I can just read the question if you want.
Yeah, sure.
So thank you for a fascinating talk. The correlation with prognosis is very interesting. Besides cell-intrinsic effects, did you see if immune infiltration was increased with chromotripsis?
Interesting question. So we didn't directly look. So we have characterized the degree of immune cell infiltration using the RNA-seq. So by doing bulk deconvolution and also doing deconvolution using some single cell data that we've got. Whether we correlated that with the presence of chromotripsis, I'm not sure. That might have been done, but not by my group. That might have been done by Colin Semple's group. If you're particularly interested, you should drop him a message. But to my knowledge, we didn't see anything particularly exciting there.
Thanks a lot.
Stefano, do you want to ask?
Yeah, hi, good afternoon.
Hi.
Thanks for the interesting talk. I've been wondering whether you have a mechanistic idea of how structural variation and copy number alteration can trigger and maintain CIN. I mean, do you see anything in terms of, or do you think there is anything in terms of DNA replication stress or other type of features that might fuel this vicious cycle?
So more broadly, I don't know. In the simplest case, it seems feasible. So where you're thinking about particularly deletions, the loss of genes, where you're losing the function of BRCA1 and 2, where you know that loss of function of those genes results in DNA repair deficiency, which then we know results in characteristic scarring and etc., results in chromosomal instability. You can kind of see how that might be happening. For more, in my head, more complicated mechanisms, or where the mechanism is harder to understand exactly what's happening, I don't think there's this clear hypothesis of exactly which changes would be inducing that, or where you would start to look at that. I think that's a really interesting question. And I'm not sure if others are looking at that. I would be really interested to know if they are.
Thank you so much.
Any more questions? Doesn't seem to be the case. So yeah, thank you again for this fantastic seminar. So our next lecture will be after the Christmas break, so on the 16th of January. So we will be hosting Tisha de Lang. So we'll start the year on telomere biology. And yeah, thank you very much, everyone, for joining, and enjoy the rest of the day.